Well, thanks everyone. Okay, well, thanks everyone for coming. It's my pleasure to introduce Catherine, Danielle, and Marion, who are all University of Calgary students, and they are all part of the university's International Genetically Engineered Machines competition team. And so this is a competition called iGEM. It's run every year. It's a global competition that asks teams from around the world to design biological solutions to all sorts of different problems. And so this year's University of Calgary team has come up with a really innovative way to use biology to try to tackle the problem of produce spoilage. And so I won't go into too much detail about their project. I'm going to let them explain all of that to you. But the team did just come back from Paris, where they were at the global finals of the competition, and they won their track in the competition. So that's a really big accomplishment, especially for a Canadian team to come first. So I'm going to pass it over to you guys, and I'm sure everyone was really interested to hear more about your project.
Yeah, thank you, Emily, for the introduction. As Emily mentioned, I'm Marion. I'm joined by Danielle and Catherine. And today we'll just be presenting a little bit more about our project. Again, as Emily mentioned, we are part of iGEM, which is the Internationally Genetically Engineered Machine, which is a competition that really encourages advancements in synthetic biology, education, and collaboration within the scientific community. And beyond just synthetic biology, iGEM also encourages teams to really participate in education initiatives, interact with professionals, and consider entrepreneurial angles to your project. And for those of you who may not be familiar, synthetic biology which is really the foundation of iGEM, is really a field dedicated to redesigning organisms for novel applications in engineering. So it leverages biological systems like bacteria and allows us to modify them to make specific solutions. So for example, for our project, we engineered bacteria to produce bioplastic as well as an antimicrobial peptide. And with that little bit of context, we can jump right into our presentation. So Canadians love fresh fruit, but our landfills do not. Each year, Canada loses over $50 billion in avoidable losses due to food waste. However, this problem is all over the world. Fruits and vegetables represent 45% of global food waste, and the environmental and economic implications of this are substantial. Before we started developing a solution, our team reached out to stakeholders within the packaging industry to better understand the problem and what kind of solutions would be appropriate. After speaking to Chris Clark, the category director at Star Produce, we learned that Canada's food waste problem is unique. Because the majority of our fresh fruit has to be transported long distances, the harvesting, packaging, shipment, and distribution pipeline can take up to several weeks. Stone fruits like peaches have delicate skins and limited shelf lives. This means that the long transportation times leave them even more susceptible to rotting and bruising. This cuts short the time that produce is edible before it needs to be thrown out. And we also learned that current preservation methods cause significant losses in quality. So our conclusion was that we needed a better, longer-lasting preservation solution. Before we could really dive into the design of our project, we also had to acknowledge that food waste is not the only problem we had to face. The food industry is heavily reliant on single-use plastic packaging, which also ends up in our landfills and has substantial consequences for the environment. In June of 2022, Canada joined 77 countries' efforts to reduce or ban single-use plastics. Based on interviews with consumers, we also found that people generally want to see less plastic on their food. Together, these factors represented a really pressing need for sustainable alternatives to single-use plastic. Based on these findings, we knew that addressing the problems of food waste and plastic waste were urgent. And so we thought to ourselves as a team, how can we use synthetic biology to prevent spoilage and make an affordable plastic alternative packaging? This is where Cellucoat comes in, a packaging that keeps produce fresh from the field to the fridge. Our vision was to use synthetic biology to deliver a sustainable and antimicrobial plastic alternative to prolong the shelf life of produce, helping people both reduce plastic and food waste. Through an iterative design process, we developed Cellucoat with three main components: preservation, production, and protection. Let's take a closer look at the details of our project.
So since one of the primary goals of Cellucoat is to extend the shelf life of fruit and vegetables, we asked ourselves, what is the root cause of spoilage? In our meeting with Josh McGinnis, who is a fungi and bacteria expert, we learned that produce is vulnerable to a wide variety of pathogens, both during transport and at the store. So we went to our local grocery store and we collected samples of rotting fruits and vegetables. We swabbed their surfaces and grew the fungi and bacteria on plates. And with the help of Josh, we were able to identify which species were present. We knew that if we could identify an antimicrobial peptide that targets these pathogens, we could reduce the effects of spoilage. But when we searched for an antimicrobial peptide in literature, over 880 different results came up. This was not a haystack that we could easily sift through. So to refine our search, our team designed an involutional neural network, which allowed us to automate and complete the screening of antimicrobial peptides in a matter of minutes. Using our new software tool, nisin came up as a key hit. Nisin is an antimicrobial peptide that targets gram-positive bacteria by binding to lipid 2 on the pathogen's membrane, creating a pore and leading to cell death. Nisin is generally recognized as food safe by the FDA, which was an integrated design principle that's necessary to our application as a food preservative. As well, it is also pH and temperature stable, meaning that it can withstand long transportation times. Natural sources of nisin, like the L. lactis bacteria strain, make it difficult for mass production of nisin at an industrial scale, which is why our team designed a part collection for nisin Q expression in E. coli. But after cloning our first genetic construct, we ran into a problem. We hypothesized that our protein was difficult to secrete and too small to visualize on a gel. So we created a second genetic construct and we added NUS-A as a soluble factor to increase peptide in E. coli, but we still did not produce a band where we wanted. So we went back again and we improved our design by adding both NUS-A and GST as secretion and purification tags, and we were able to successfully produce nisin protein. Now, thinking back to our interviews with industry stakeholders, because the transportation process can take up to several weeks, our application for nisin needs to be active for long periods of time. So to characterize nisin, we designed a new measurement tool called KB-Perry that standardizes Kirby-Bauer antimicrobial tests by producing more precise zone of inhibition measurements. We tested nisin's effectiveness against B. subtilis, and KB-Perry validated that nisin is an effective preservative against gram-positive bacteria even after fruit begins to spoil.
With the ability to produce an antimicrobial peptide, our team had to figure out a material that we could integrate into produce packaging. We initially turned to bioplastics, but after an interview with Natalia Gonzalez from the Calgary Waste and Recycling Services and Belinda Lee, an environmental engineer, we understood that even though a material is biodegradable, if it can't decompose in city compost facilities, it will be thrown into the landfill. So our team looked into materials that honored a circular economy design where it could be created from the very products I would break down into. Our search narrowed down to bacterial cellulose, or BC, produced by xylinus. It's a pH and thermostable polymer of glucose molecules that is regarded as safe by the FDA for consumption. We approached Chris Clark again with our idea to use BC as a packaging material. He asked us if our material could be used for different packaging purposes, as produce needs to be packaged in bags, boxes, or wraps. So we decided to test flexibility and strength through uniaxial tensile testing on our BC, and we found it was similar in strength and flexibility to paper, meaning it was 3 times weaker in both respects compared to plastic. On its own, BC wouldn't quite cut it. If Cellucoat wanted to be a viable replacement for plastic, we knew it needed to be versatile in packaging types. So our team once again turned to synthetic biology for a solution.
The iGEM competition is no stranger to PHB, a biopolymer that has been recombinantly produced since its creation by the 2012 Tokyo Tech team. However, our team was drawn to PHB because of its potential to enhance the flexibility of our BC. It's also compostable, biocompatible, and produced from renewable resources, which aligns with our project's core value of renewability and sustainability. We wanted to be able to use the existing PHB genetic construct designed by the Tokyo team and make a platform that allowed us to fine-tune the amount of PHB incorporated into our BC. This would allow us to customize the mechanical properties of our packaging material. While there are 4 proteins that are important for the PHB production and secretion process, we zoned in on a protein called phasin, which coats PHB granules inside the cell and prevents them from aggregating into large clusters. And this is also really important for secretion. Because of phasin's role in regulating the amount of PHB that's secreted, our team created 3 different strengths of ribosomal binding sites, or RBSs, upstream of the phasin gene. This The different RBS strains would change the amount of phase and protein produced. In turn, this would increase the amount of PHB that was secreted into our material. Based on our results in the lab, we saw that changing the strength of the RBS did result in an evident change in the amount of phase produced. So with our platform created, we once again attempted to test the flexibility of a prototype PHB-BC composite containing both materials, and we found that it was 6 times more flexible than the original BC. This means that we could change the properties of Cellucoat for different packaging purposes, like increasing PHB concentration to make a stiffer material for boxes, or decreasing it for more flexible materials like wraps and bags. Cellucoat now had the ability to theoretically replace both hard and soft plastics using a single renewable and composting material. However, another meeting with Belinda Lee brought up some concerns about how, even though PHB and BC are compostable on their own, the combination of the two materials would potentially compromise this compostability. So we once again reached out to the Calgary Waste and Recycling Services to test if our PHB-BC composite material would break down within the industrial composting cycle. And after one composting cycle, Natalia Gonzalez got back to us and showed us our results. While bioplastics still have a long ways in general before they'll be accepted into industrial composting, our material degraded substantially and looked similar to paper and had like a natural appearance after it had biodegraded. These results were promising for the future compostability of our material. And as citywide composting facilities come to expand their capacities, Cellucoat will thus be able to be a material that's both sustainable from creation to degradation.
Now our project has 3 parts, 2 E. coli strains producing either PHB or nisin and xylinus producing BC. The only thing left was putting nisin, PHB, and BC all together to make a single packaging material through a streamlined process and holistically tackle food spoilage. So we worked out a coculture method where we would grow all 3 types of bacteria simultaneously to streamline Cellucoat's production process. However, Dr. Hu, a bioengineering professor, warned us that cocultures were very unpredictable, and understanding and controlling the environmental factors that affect growth is key. So we created a mathematical model capable of optimizing coculture conditions to get the greatest possible BC, nisin, and PHB yield. The result of this model would then inform the design of the experimental coculture conditions, such as the initial bacterial concentration, growth conditions, and growth-limiting factors. Docking simulations also supported our decision to use a coculture, as we discovered that nisin has strong interactions with BC and PHB without interfering with its antimicrobial activity. We also used molecular dynamic simulations to look at nisin's behavior in high or low temperatures and pH. This confirmed that nisin's affinity to BC won't be affected by different environmental conditions, and it can protect fruit through wider transport conditions.
Now that we had a process to streamline BC, nisin, and PHB production, we asked ourselves, how can we distribute our proteins into the cellulose fibers to make sure that every part of our packaging was protecting the fruit inside of it? Because protein secretion from E. coli is difficult, we met with Dr. Birkinshaw from the University of Calgary, who inspired us to use our purifying process to our advantage. By using thermolysis, we can both kill E. coli and release our proteins into our bacterial cellulose packaging, creating an antimicrobial and consumer-safe product. We tested our thermolysis technique on GFP-expressing E. coli to visualize how well this process worked. Based on our results, we saw that even after sterilization in the autoclave, our BC sample that was grown in a coculture with E. coli expressing GFP was still fluorescing. This means that even after autoclaving, that our protein can remain in our bacterial cellulose packaging. In our final coculture design, we planned for xylinus to produce bacterial cellulose, and E. coli will be added to the coculture. E. coli containing PHB or nisin will become distributed throughout the fibers as they form. E. coli will begin to secrete PHB, and using our thermolysis approach, we can efficiently release nisin and the rest of PHB into our packaging material. So while we know that our packaging works in a lab, it doesn't necessarily mean that there will be a market demand for it. So we decided to meet with experts of the food packaging industry. Our first meeting was with industry contact John Kelly, who specializes in the produce and market and sales. He showed us that alternatives to plastics need to be cost competitive, and unfortunately, a lot of sustainable products often fall short in this category. When we conducted our own cost analysis, we found that BC production is very expensive. So to combat these costs, we created a growth media using recycled fruit waste. And using this, we can reduce production costs by a whopping 60%. According to uniaxial testing, our fruit waste media won't compromise the strength of our bacterial cellulose either, allowing us to create a price-competitive product without sacrificing quality.
However, price isn't the only thing that consumers are concerned with, so we went back for another meeting with Chris Clark to make a more customer-friendly packaging. Chris stressed that customers are visual shoppers and prefer packaging that they can see the product with, while retailers gravitate towards packaging that has a tint to conceal aesthetic deformities on the produce. So we developed a sodium bicarbonate purification method that improved the transparency and strength of Cellucoat. We also consulted trend researcher and biodesigner Juliana Schneider, who recommended that we use natural dyes such as cabbage juice to give our packages a natural artesian tint without compromising compostability. Lastly, our packaging material has to be able to be made into a package. We used a process called biosculpting to dry the BC in various 3D shapes. Cellucoat has transformed into a product capable of helping people worldwide save money on reducing food waste. But to do this, we need to see if it's competitive to other packaging solutions. After conducting a competitive analysis, it quickly became evident that Cellucoat exceeds in every area that makes a packaging desirable for both consumer and buyer.
So through the use of synthetic biology, our preservative, productive, and production qualities make Cellucoat a better food packaging. The future of our product offers customizability in a variety of ways, all through a single system. We can target fruit-specific pathogens by identifying new antimicrobials with our neural network. We can replace both soft and hard plastics through fine-tuning RBS strengths and producing different levels of PHB. And we can easily change color and shape to meet consumer needs. It has been a journey, and thanks to our wonderful mentors, industry, and HP contacts, Cellucoat is better equipped to keep your fruit fresh from the fields to your fridge. So thank you so much to all of our sponsors and our funders, as well as our principal investigator, Dr. Mai, who have all just been a really big help to our project throughout the year. And yes, thank you all for listening. If you'd like to have more information on our project, we did create a website for the project, which I think is just there. So yeah, we'll now take any questions if you have any.
I have to say how absolutely amazing and complicated this work seems to have been. You approached so many different people to make sure that different aspects of it work. I must say I don't understand the details of it, but the objective seems to be extremely beneficial. So I don't have any questions immediately, but I was just mentioning that while other people have a chance to either submit some questions or jump in and ask a question.
Thank you so much. Yeah.
Perhaps I could ask a question, Hugh. I'd like to congratulate you first. It's an amazing product. Let's hope it works. So I have two questions. One was, how long did it take you to get through all of this? It seems like an awful lot of work.
We started this project in a small group in our winter semester, so beginning like February, March, but we really started in the lab in May. So we've had the summer and spring. And that's kind of it. We were very busy.
Yeah, it's more than I've seen some masters or PhD students produce. So I'm hoping you get a better degree than just a BSc. So the second question is a bit of a strange one. So if you protect all the fruit and vegetables so that they come to us fresh, where are you going to get the rotting fruit from that forms your medium?
Yeah, that's a great question. And what we noticed when we really spoke to these industry contacts is that food waste is endemic. So if you think about fruit that's being picked or like apples that fall off the tree, there's always going to be fruit that isn't meeting that quality that consumers want. And there's already systems in place, like they already send that to produce like dog food and other things. So it would just be working with that and leveraging that. And unfortunately, that's always going to be a thing. But yeah, great question.
Yeah, and the last thing then is, have you had any ideas about how to commercialize this?
Yeah, I can jump in again. So beyond our competitive analysis and cost analysis, we're really just talking to members of the packaging industry. And like we mentioned, there really is a demand for sustainable alternatives. So moving forward, we would look into like patenting our solution, and then perhaps like expanding tests and stuff like that. But it would definitely involve more work with people in the industry and also talking to customers and consumers to refine the design and things like that.
Yeah, so thank you very much. I mean, great, and I hope you're really successful. All the best to all three of you, or all the whole team anyway.
Thank you very much.
So have you, is it patented or have you protected your idea in some way?
So with iGEM, we're really encouraged to begin the process of applying for a patent. It hasn't been patented yet, but yeah, I guess that's something we're working towards.
So my next question really is related to that. Would it be easily duplicated? Could other people do it? And I mean, it's surprising to me that you've been able to do it in 7 or 8 months, but imagining you had a professional company doing it, would they have been any quicker, do you think?
Well, so using bacterial cellulose for like other applications, like it's used in medication as like bandaging material and stuff like that. So there is already work with that. Our specific application of adding nisin and PHB, I think, is unique, although PHB has been added to BC separately, and that was kind of our inspiration. Would they be able to do it faster? Possibly, probably. We're a group of undergraduate students,
right.
But the idea of iGEM is really just like coming up with a new idea and like running with it. So.
No, I think it's amazing. My last question is really a mechanical one, and that would be, how thick is this material? Is it like cling film, or how would it compare?
Yeah, I can. Oh, go ahead, Ahmed.
Nah, the BC itself is around, it's just fractions of a millimeter. I think the thickest that we've produced into like a box is about like 0.5 millimeters, like 0.3 to 0.5. So it's compared to like plastic film, it's probably like an order of magnitude thicker.
Okay.
Plastic film is quite thin.
Right. Would that be a drawback for when you're talking to the commercial people, or would they see it as an advantage?
I mean, considering we're not really competing with plastic film with this current iteration of Cellucoat, we're competing more with plastic clamshells.
Okay.
The thickness is a lot less of an issue to those particular suppliers.
Thanks very much. It sounds like you've really looked at this from a number of different angles, including the commercial or the end use, which is very rare for an undergraduate project. Amazing.
I had one quick question regarding application. So how do you envisage sort of application, applying it sort of on a larger scale? Have you looked at that?
In terms of scaling up production, do you mean?
Yes.
So in terms of scaling up, that would be a little bit of a next direction, I guess. We have looked at like the cost analysis at a lab scale. In terms of scaling up, if you think about what bacteria really needs to grow once you have these bacterial strains, it really all it needs is the media and like incubation and a container to grow in theory, and then you can shape it. So theoretically, it should be possible. We'd have to look a little bit more into like what kind of labs look like right now that are doing more industrial levels of production, like for those biomedical applications. But yeah, definitely something to look into.
And yes, I'll just add a bit to Marian. So in addition to all she said, we have looked at, we've conducted like cost analysis and a bit of techno-economic analysis on how the production would look like when on an industrial scale. So we've compared the total, we've done a financial analysis totaling how much this will cost and the specific materials that we would need to scale up. So it is feasible. The only drawback is that it's still more it's more costly to produce BC at a large scale compared to plastic. So that's where our media comes in.
Okay, thank you very much. Emily, are you still on, or? She mentioned she was at an airport, so she may be traveling. So
we had two great presentations, Hugh. So we should do this more often. Have a speaker council.
Yeah, well, I'm thrilled at who's been the speakers. It's been wonderful to hear the different topics, and maybe I can just wrap up by, unless there's any other questions, I'm just going to say thank you very much indeed, Catherine, Danielle, and Marion, and the other speakers who jumped in there. So with that, I think we will just say thank you and very much appreciate your sharing your research with us. And again, Ron, to you as well, thank you. And I think we'll stop the recording, Nigel, and then shut down the evening.